missing. So just so this is on my recording, I didn't start the recording until after I've been through a couple slides. So go back and review the functions of meiosis. Okay. Does everybody have this? Yeah. So this is the human life cycle. And what I want you to notice here, here's the guy, here's the woman. Meiosis, like I said, it occurs in the ovary and the testes, gives you gametes. Now, fertilization occurs, egg and sperm join. Then you get the zygote to the embryo to the baby, but your zygote undergoes mitosis, cellular reproduction, making exact identical cells. So that's how we grow. Remember, we talked about growth and development is mitosis. So the rest of your life cycle is mitosis, mitosis. The only part you get meiosis is the production of egg and sperm. Now, the interesting thing about women is we are already born with all of our eggs to a certain extent, but our eggs have not fully undergone meiosis, right? And so when we hit puberty, the egg will finish off meiosis and then you go through ovulation and it's released. Okay, so another important piece of information to remember about meiosis is the difference between haploid and diploid cells. Okay, so I always think haploid half and diploid di means two. So a haploid cell has a single set of chromosomes, whereas a diploid has your full set. So if you look down here on the bottom, first of all, we symbolize diploid as 2N. We have two pinks, two purples, two yellows. So if this is your body, you have one pink from mom, one pink from dad, one purple from mom, one purple from dad. Haploid means you have a single chromosome per what will be a pair in diploid, right? I have one pink, one purple, one yellow. Now, the only cells in your body that are haploid are your gametes, right? You have a haploid sperm, haploid egg, they undergo fertilization, and you end up with a diploid zygote, a zygote with its full set of chromosomes. Why is it important that our gametes are haploid?
So we could combine?
Yeah, exactly. The sperm and the egg each have 23 single chromosomes, but we, in all of our cells except our gametes, have 46 single chromosomes. So 23 from the sperm, 23 from the egg, they fertilize, now you have 46 again. Does that make sense? Now, two other terms I want to give you here are somatic cells versus sex cells. So somatic cells are like your regular body cells, right? All the cells in your body except the ones that will make your gametes. Your sex cells undergo meiosis to produce gametes. Does anyone need more time? Nope. Okay.
Yeah.
Oh, sure. Okay, do you have it?
Yeah.
Okay. So this should be a reminder by now, but homologous chromosomes are not identical. They carry the same traits or genes, right? But you could have different alleles. Whereas sister chromatids are identical. They occur after S phase of the cell cycle and they are joined by the centromere. So the purples are identical, the greens are identical, but the green sister chromatids are homologous to the purple. Does that make sense to everyone? So as I go through meiosis, I have a bunch of different videos that I think are useful for you to visualize the process. So hopefully you're not going to get sick of videos, but I think they really help. All right, so your learning objectives: identify the number of daughter cells formed during meiosis, whether they are haploid or diploid, and whether they are identical or genetically unique. Describe the processes that occur during each stage of meiosis and whether meiosis 1 or 2 is more similar to mitosis. Oh. All right, so a few things I want to point out here. Don't worry about this reduction division. We'll talk about it later. Actually, all it really means is the number of chromosomes per cell is reduced by half. And so in meiosis, the cell is going from diploid to haploid. That's what that means, right? You're going from 46 chromosomes to 23 chromosomes. So a couple things I want to point out here. Meiosis has 2 rounds of cell division, meiosis 1 and meiosis 2. I'm going slow here because this is really key to understanding meiosis. Okay, so 2 rounds of cell division. So here we have the diploid parent cell. Now this is after S phase. How do you know it's after S phase of the cell cycle? Remember, it's part of interphase, G1, S, G2.
Is it because they segregated?
Well, not quite yet. What do you guys see here with the centromeres? What do you have there?
They went to the opposite sides of the poles.
Well, not quite yet. The
They
key
have-
here is that you have sister chromatids. Remember, sister chromatids are formed during DNA synthesis or S phase. So DNA replication or synthesis does not occur during mitosis or meiosis. It occurs during interphase. Does that make sense?
Mhm.
Okay. So in our first round of cell division, and this is not showing you all the phases here, what I want you to see is that you go from one parent to two daughters. Now, that starts meiosis two. Each daughter cell undergoes division, and then you end up with 4 haploid gametes or daughter cells. So I'm going to write that down for you. So 4 genetically unique because we get genetic recombination. Wait, divided into 4 genetically unique daughter cells. I don't think I needed to put a comment there, but whatever. Oh, yeah, 4 haploid genetically unique. Knew I was missing a turn. There you go. 4 haploid genetically unique daughter cells. And when I say genetically unique, I mean different combinations of alleles. They all have the same genes, but you have different combinations. Does everyone have this? Okay. So we're going to do an overview. Hold on, I got to enable my audio. Sorry about that. Okay.
Happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into 2 stages of cell division, called meiosis I and meiosis II. Meiosis I has 4 phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, 1 from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase II, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase II, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with four genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has two stages of cell division called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce two haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces four genetically different haploid gametes. And here are some key points to remember about prophase I. The pairing of homologous chromosomes called synapsis occurs. Each pair of homologous chromosomes consisting of four chromatids is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
Okay. I like that video because it gives you a good way to visualize meiosis. So let's go.
Miss Dr. Foltz, is that video part of the, is that in one of the modules too? I don't recall.
It is in the Google Slides, and I cannot remember if I added it to the instructional videos. I can do that though if I didn't already put it there. Let me make myself a note. But yeah, it is in the Google Slides. Which, to make it work, you know, to make the link work, you have to put it in slideshow. All right, so meiosis 1, meiosis 2. Everything that occurs in meiosis 1 is occurring to homologous chromosomes, right? Homologous chromosomes line up in metaphase. Homologous chromosomes are pulled apart in anaphase, whereas everything in meiosis 2 is happening to sister chromatids. When I say everything, I mean all of the processes during the phases. Okay, does everyone have this? All right. So in this image, it tells you what happens during each phase. Chromosomes condense, nuclear envelope breaks down, crossing over occurs in prophase 1. Metaphase 1, pairs of homologous chromosomes move to the equator of the cell. Think equator, middle. Okay, so you can go back and write this information down. So meiosis 2 is similar to mitosis, but we'll get into that later. So I put here, does crossing over occur during prophase 2? So before we answer that, I want you to look at this slide here. So this is crossing over. So it occurs during prophase 1 of meiosis, and it is crossing over that leads to genetic recombination or a unique combination of alleles. Does everybody have this? All right, so I'm going to write a few things down, which hopefully will help you understand crossing over. So let's take a look at these homologous chromosomes. We have blue sister chromatids. They're identical. Notice we have gene A, gene B, gene C. For that gene, blue is the dominant allele, dominant allele, right? Red has the same genes, but they're all recessive alleles. Okay. So I'm going to write this here. Let's say gene A is, actually, I'm going to take that blue off. Gene A is hair color. Okay, so we have capital A equals brown, lowercase a equals blonde. Gene B is eye color. Remember, eye color would be the trait. Let's say capital B equals brown eyes. Lowercase b equals blue eyes. Gene C equals height. Capital C equals tall. Lowercase c equals short. Okay, so I'll let you guys get that written down. Savannah, I'm recording this. I see you're driving, but I'll post the recording. You don't have to answer, I see. Okay, so our blue chromosome, that's an abbreviation. You have brown hair, brown eyes, tall. Okay. See, because the blue chromosome is capital A, capital B, capital C. The red chromosome, whoops, you have blonde hair, blue eyes, short. So what this is referring to is alleles, right? On your blue chromosome, brown hair goes with brown eyes, goes with tall. A red chromosome codes for blonde hair, blue eyes, short. Does that make sense to everyone? So now if you look here, during crossing over, parts of the non-sister chromatids are exchanged. What I mean by non-sister, see, the reds are sister chromatids, the blues are sister chromatids, but you're taking a chromatid from the red and a chromatid from the blue, and they're exchanging alleles. So they're still chromatids, but they're not sister chromatids to one another.
Dr. Folt,
So,
during crossover, does it matter if one gene is recessive and the other one is dominant?
No, that's a good question. You mean, is that where it will definitely cross over?
Yes, like it'll take the more dominant gene to cross over instead of the recessive, or does it matter?
No, it doesn't matter. And so that's a good question. Crossing over occurs all up and down your chromosome, right? We're showing it for just one gene, but it actually occurs all up and down for all these different genes. And it doesn't matter whether one is dominant, one recessive, it's just crossing over. So it may be like, recessive crosses over with recessive.
Oh,
Does that
okay.
make sense? That's a good question. Okay, so we have crossing over. And so now, according to our picture, right, we're looking at the blue chromatid here, the red chromatid here. The gene for height, they're exchanging alleles for the gene for height. So now your blue chromosome will have this combination: brown hair, brown eyes. But if you look here, a capital C switched with a lowercase c. So now you're short. Whereas the red chromosome, you have blonde hair, blue eyes, tall. So this is a new combination of alleles. Now you have a sister chromatid that has brown hair, brown eyes, short, whereas originally it was brown hair, brown eyes, tall. Same with the red chromosome. It was originally blonde hair, blue eyes, short. Now it's blonde hair, blue eyes, tall. Does that make sense to everyone? We're going to watch a little video on crossing over as well. We'll let everyone get this written down.
So is tall a dominant gene? Or was that just for an example?
It's just my example. And actually, everything that has like a continuum, right? Like height, you could be 5'3", 5'4", 5'5", right? It's not just tall, short. Like eye color, you could have all different combinations. Like I have hazel eyes. All of those are coded for by many genes, and those genes interact with each other. So for instance, eye color, I was just looking this up, is coded for by 15 genes on different chromosomes. And so there will be a gene that says pigment, no pigment, right? And then if it says pigment, all of these other genes will work together to determine the pigment. And that's even a simplification, right? It's really crazy. Like if you have two blue-eyed parents, you can still get a child with brown eyes, green eyes, whatever. So it's pretty interesting.
Is
And
that
like I
because
said,
parents- Is that
Okay.
because the grandparents may have had the more dominant brown eyes?
It could be, or there are subtle changes in the expression of the genes,
Oh,
right? With epigenetics, which we'll get into next week. So epigenetics, it controls like when genes are on or off, right? And so but it's like environmental factors. It's not mutations. And so maybe there's some environmental factor that turns that pigment gene on, right? So it doesn't necessarily have to be in the grandparents. Like a lot of times they say it skips a generation. It's not quite how it works. It can work that way, but not always. Yeah, it's pretty cool. Okay. I think nope, where's my crossing over? It's my independent assortment. I don't want that yet. I have a good crossing over video somewhere. I think this is it. Actually, I think this is the same video I linked earlier. So let's just watch the crossing over part again.
Introduce
I'm going to skip up to the crossing over.
After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete.
Okay, so like I said, crossing over is the exchange of alleles, but it's for the same gene, right? That's how you can get eggs that all have different combination of alleles, or sperm with all different combinations. And so you could have, say, 3 kids with your partner, and they're all going to have a slightly different combination of alleles. Okay, so the other thing I want to show you is independent assortment of homologous chromosomes. So this occurs during metaphase 1. So crossing over only occurs in prophase 1, because prophase 2, you no longer have homologous chromosomes, right? Now, the next place that you get genetic recombination is metaphase 1, independent assortment of homologous chromosomes. So let's look at this, possibility 1. So we have red long sister chromatids homologous to the blue, red short homologous to the blue short, right? They're lined up on either side of the equator. The equator would be going right through the middle here. I'm going to draw a line to help you picture that. Okay, so here is the equator. Now you get the general idea. So we have long red, short red on one side, long blue, short blue on the other. So when the cell divides, remember it's going to divide along this equator or metaphase plate. You get long red, short red in one daughter cell after meiosis 1, long blue, short blue in the other daughter cell. Okay, so now the equator is going right through here, right through here. And so now this is where your daughter cells will divide. You get a long red, short red, long red, short red. And so these would be your eggs or sperm. Okay. So if you look here, you have little a, little b. Those are together. But now let's look at possibility 2. Now we have red long, short blue. Now remember, these short ones are homologous, the long ones are homologous. So you're still getting the same genes in all of your daughter cells. So now after meiosis 1, remember your metaphase plate or equator is going right through there, right through there. That's where you get the cleavage furrow and the separation of cells. But now you have a very different combination in your gametes. You have little a, big B, instead of little a, little b. So again, genetic recombination. Your gametes have a different combination of alleles than the parent cell. And they're all genetically unique. So genetic recombination occurs during prophase 1 with a crossing over. and metaphase 1. Random or independent, same thing. So like the video said, crossing over is random. Where those non-sister chromatids exchange alleles is random, right? The genes are like randomly selected. The same is true of independent assortment of homologous chromosomes. Whether it ends up with red and red or, red and blue is random. And if you picture in the sperm cell or the egg cell, you have 46 different chromosomes, right? And so there's all different possibilities. I'm just showing you 2. Does everybody have this? Okay, I want to go over one other thing, and then I'm going to show you how to do the homework. So we're going to skip down here to problems with meiosis. Now, Down syndrome is caused by what we call trisomy 21. And so what happens here is the 21st chromosome, right? You should have 2 representations, one from mom, one from dad. But what happens is called non-disjunction. And so the chromosomes do not separate correctly during metaphase or anaphase. And so the resulting, usually it's the resulting egg has an extra 21st chromosome, right? And then that has all kinds of implications for the development of the baby. So let me show you how this happens. So here is meiosis 1. Remember, homologous chromosomes are separated, but during non-disjunction, there's an accident, right? And the homologous chromosomes are not separated. So now, during meiosis 2, you have one cell that just has these little chromosomes here, right? Because those separated like they're supposed to. But then these did not separate. So now your resulting daughter cell has 3 chromosomes in each, like on each side, rather than 2. And this one only has 1. So you end up with gametes. This is called N plus 1. So N means haploid. The plus 1 means an extra chromosome. So we have N plus 1, N plus 1. So if these were, say, eggs, 2 eggs have an extra chromosome of chromosome 21, if you're talking about Down syndrome. And 2 eggs and minus 1 are missing a chromosome. Now, you can also get non-disjunction during meiosis 2. And so what occurs here is sister chromatids do not separate. So in one daughter cell, you have the normal number, right? Because in this one, the sister chromatids did separate, so you get your normal gametes. But in this one, sister chromatids did not separate. So you get an N plus 1 and an N minus 1. Okay, so non-disjunction, either homologous chromosomes do not separate, or in meiosis two, sister chromatids do not separate. And depending on whether it occurs in meiosis one or meiosis two, that determines like how many of your gametes would be normal. Does that make sense? And so why does this happen? Well, in the case of Down syndrome, there's not just one cause, but typically, as women age, the eggs, remember when you ovulate, or when you begin the menstrual cycle, your eggs finish meiosis, right? I think they're arrested or stopped in like metaphase one. And so then when you're ready to ovulate, the egg goes through the rest of meiosis. And when I say egg, I actually mean the one that goes through meiosis. So in women, we have what's called an oocyte. And so the oocyte is the one that will then undergo meiosis and give you 4 eggs, right? Okay, the oocyte undergoes meiosis and gives you 4 eggs. So when women are ready for, the oocyte to mature and undergo meiosis, as you age, it doesn't work. There's the chance that it won't work correctly, right? So I think if you get pregnant and you deliver by age 35, you run a slightly higher risk of this happening. I think it's still really low, but it's slightly higher. So in males, it's called the spermatocyte, and that's what undergoes meiosis to give you 4 sperm.
Can like male increase the chances of that happening too, or is
Yeah.
it just female? So
That's a good question. Yes. It can happen in the sperm. And so when sperm are produced, they can have an X or, I'm sorry. Yes, it can happen on the 21st chromosome as well. And there's all kinds of different things that can happen to chromosomes like in sperm. And so that's what this homework assignment is going to show you. So let's jump over there. I know I only have a few minutes left. So you guys are going to do this meiosis game. And so read the disclaimers, right? This game assumes that the mother has 2 X chromosomes, father has XY, even though it doesn't necessarily work that way. Okay, so here we have mom and dad, right? And so let's say this is the spermatocyte. It will undergo meiosis to give you sperm, right? Now remember, the sperm have all your chromosomes, 23, all the genes, right? Okay, so here we are in prophase 1. So remember, DNA synthesis or replication occurred. Now I am going to select disjunction in prophase 1. That means that the homologous chromosomes separated like they are supposed to. Okay, so everything's good. And so this is just looking at sex chromosomes. That gives you an X chromosome with sister chromatids and a Y. But let's say in mom, she's XX, we have non-disjunction. Separation did not occur correctly. So now, after meiosis 1, you have a daughter cell with 2 X chromosomes and a daughter cell with none. Okay. Now the one that has no chromosomes, it does not undergo division again. So now let's say mom in prophase 2, she undergoes disjunction and disjunction. So now they're dividing correctly. She ends up with 2 eggs, each of which has 2 X chromosomes. They are not haploid, they're diploid, right? And you don't want that. So let's say dad undergoes disjunction and disjunction. So his sperm are all haploid. So now select one gamete from each parent. So here's mom's egg. And let's say dad's sperm, we'll use the X. You end up with an embryo that has what we call triple X syndrome. 47 means there's an extra chromosome, right? Because normally we have 46, and they are XXX. And so this tells you what happens in a person who's triple X, right? So it says here, condition only occurs in females. They have an additional X chromosome. How the person is affected will depend on the proportion of triple X to regular cells. Okay. So what this is supposed to help you understand is, first of all, the gender continuum. How do we get people that do not identify as male or female, right? And a lot of times this has to do with their sex chromosomes, but not always, right? There's a ton of different chromosomes that code for hormones and all that kind of stuff. And so this whole exercise, let me go back to the document, will help you understand that. Now, I give you what I hope are very clear instructions on how to do this. So, non-disjunction during prophase 1. Trait number 1, select the following for the mother. Prophase 1 disjunction, prophase 2 disjunction. Select the following for the father. Prophase 1 non-disjunction, prophase 2 disjunction. So let's take a look at this. We go over here. I'm going to start this over. Somewhere. Where's my clear button? I don't know where my clear button is, but we'll do this. That didn't work. Huh, weird. All right, well, let's just do it over. So it says mom, select disjunction. Oh, that's all you do is just reselect. And then I told you disjunction, disjunction. So mom has 4 normal eggs. For dad, I said non-disjunction for prophase 1. And then I said disjunction and disjunction. So here's what dad ends up with. So then I said, pick a gamete from mom, a gamete from dad, and look at the offspring. Okay, does that make sense to everyone? Now you're going to go back here and you're going to fill in this table. So trait number 1, here's what I told you to do for trait number 1. That's what we just did. You're going to put in the karyotype. It was 47, XXY. Name of the trait, it's Klinefelter syndrome. How common? So you look down here, you have to read all this stuff, and it says here, somewhere in here, it occurs in, here we go. Roughly between 1 to 500 males, 1 to 1,000, right here. Okay, so that's what you put in for how common is it? And then I ask for 3 physical aspects of the trait, but for some there are fewer than 3. Okay, so just make sure that you read through the directions carefully so that you can make the correct gametes, okay? And I think I asked you to do it like 4 times. And then there's also a video as part of the assignment that I want you to watch, and it talks about like different genders. And it's not just in humans, in all kinds of animals. So, all right.
Are there questions on that too? Like
Yes,
second part of
there
the
are.
Okay.
Yep, there are. For those of you who joined late, no class on Wednesday. So review your meiosis. You have a meiosis Google Slides to do. You have this assignment we just looked at in vocabulary, no examples, and that's about it. and then a ton of videos if they help you.
Do we have
Okay.
any quizzes this week?
Yes, I put up a cellular respiration quiz that will open on Wednesday.
Okay, thank you.
Yep.
Oh, also, when I took
Uh-huh.
the test for the bacteria and viruses,
Mhm.
they said that we could use our notes on the thing. I didn't know. It says
Oh,
you are able to use your notebooks.
oops. I think
How
I
was
forgot to change the instructions,
how was
but
that?
if you,
Oh, okay.
no, I meant to change the instructions. You couldn't use your notes.
Yeah, that's what I figured. I grabbed it, but
Yeah.
I didn't open it, so
Yeah. Okay, cool. If anybody did use their notes, I'm not gonna like give them
yeah.
a 0, but
Okay, thank you.
uh-huh. Any other questions?
No, thank you for your time, Dr. Fultz.
You're welcome.
Have a great day.
You too.
Thank you.
Bye. Bye, Lorraine. Any questions?